Good morning, everybody, and welcome to the Epileptic Seizure Prediction session. I'm Dan Nguyen, neurologist and epileptologist at the Montreal University Health Center and current president of the Canadian League Against Epilepsy. So we've assembled here for you a group of excellent speakers who will provide you with an overview of the topic of seizure prediction, looking at what has been done before, what is currently being done, and what the future holds. So we will hear all three presentations, one after the other one, and the speakers will be available at the end to answer your questions. Please feel free to write your questions along the way using the Q&A function, and I will ask them to our speakers in the end. If it's relevant, please specify if the question is for a specific speaker. So our first speaker is Dr. Jean Gottman, who requires no introduction. He's a professor at the MNI, affiliated with McGill University. He's been a pioneer in the field of EEG analysis and epilepsy research for close to 50 years now. He's the recipient of a number of prestigious awards, including the Ambassador for Epilepsy Award from the International League Against Epilepsy. His talk is an overview of seizure prediction algorithms over the last 30 years. So we can start his presentation now.
Good morning. I will not give you an overview of seizure prediction algorithms over the last 30 years because it would take much more than the 12 minutes that I have available. I will just give you an example of some of the work that has been done, hoping that it will give you an idea of where things are going. The outline of my talk will discuss early work and the great hopes we had with this early work of being able to predict seizures. Then the landmark Morman paper, resetting the seizure prediction world onto the right path. A little bit of more recent work, the most important and critical NeuroVista trial, and finally conclude what can we hope for at this point. This is an example from an early 1998 paper published in Nature Medicine by the Paris group that gave us great hope. They studied 19 temporal lobe seizures in 11 patients, looking at 40 minutes of EEG around each seizure. You can see on the left, they measure the autocorrelation in the 500 seconds preceding a seizure. Time zero is the time of seizure. Nothing changed in the autocorrelation measure before the seizure. It changed after the seizure took place, of course. Now, on the right, you see the nonlinear spatial temporal measure based on chaos theory, which I will not discuss the measure itself because it's very complex. But what is important is you can see that 2 or 3 minutes before the seizure onset, some changes took place, unlike what we saw with the autocorrelation function. And this gave hope that maybe you can predict seizures, particularly because in the 19 seizures that they analyzed, you can see here, very often, most of the time, changes took place 2 or 3 minutes before time zero, the time of seizure onset. Whereas using the autocorrelation, the circles that you see here, the changes took place only at the time of seizure onset. So these nonlinear measures looked like they were very promising for seizure prediction. Another group using the correlation dimension and other nonlinear measures published very similar results around the same time. The Bond group studied 16 patients, 1 seizure each. Each patient had a 15-minute pre-ictal period, which was compared to 68 epochs of 20 minutes recorded 24 hours earlier, away from seizures. And the graph demonstrates clearly that changes in these measures occurred 5 to 10 minutes before the seizure onset. The magnitude of the change is shown on the y-axis. In a few patients, the change occurred only a few seconds before seizure onset, but again, giving us great hope that seizures could be predicted. This led to an article written by Brian Litt, who is here, and Klaus Lennertz from Bonn, where they wrote, pressing milestones necessary for clinical utility are first, demonstrating prospective seizure prediction for prolonged human data sets. Second, elucidating the mechanisms underlying seizure precursors. And three, implementing these algorithms on implantable hardware platforms. They organized the first seizure prediction workshop in 2002, and this was followed by many such seizure prediction workshops later on. It was found during the workshop that long hours, days and days of continuous EEG were analyzed, and the outcome showed very mixed results, with a generally poor performance and lack of consensus regarding the best methods and what should be the performance measure. So there was great disappointment after this early hope that in fact results were not reproducible when you analyzed very long EEG sections. This led to the paper of Frank Moorman and the Bonn group published in Brain in 2007, The Long and Winding Road of Seizure Prediction. And they wrote, while most of the studies published in the 1990s and around the turn of the millennium yielded rather promising results, more recent evaluations could not reproduce these optimistic findings, thus raising a debate about the validity and reliability of previous investigations. We will propose methodological guidelines for future studies on seizure prediction. And these guidelines are as follows. Use test data independent of training data, a lesson that I think everybody has learned today. Use long, at least multi-day continuous recordings. Demonstrate validity by showing that prediction is statistically better than that of a random predictor. So demonstrate statistical validity, not just show examples of seizure prediction. And report sensitivity and specificity taking into account the prediction horizon. In other words, it's important to state whether you think a seizure is going to occur within 5 minutes or within 3 hours. These recommendations have reset the seizure prediction world, allowing to start again on solid footing. And I think after these papers, all subsequent studies had at least much better statistical validity. This is an example of such a study in which I was involved, but it represents many studies that have been done during the last 5 or 7 years, following the criteria of Moorman. We studied 17 temporal lobe epilepsy patients, 1,600 hours of recordings, so long recordings for each patient, a large number of seizures, about 10 per patient. We performed patient-specific training, in other words, developed algorithms specific for each patient by partitioning the data into independent training and testing set. An algorithm is specific to a patient, trained on that patient's training data, and tested on that patient's testing set. The results showed that in 13 patients, we could perform seizure prediction better than chance. In 4 patients, we could not. With the prediction horizons of about 25 minutes, we reached a sensitivity of 80%. That means that 80% of seizures actually occurred within the prediction horizon. However, the patient would have to spend 25% of their time under warning. That means that 25% of the time they would be told a seizure may occur now. Now, the NeuroVista study was led by Mark Cook and the group in Melbourne. And this is really a landmark study by its magnitude. It was a prospective study of 15 patients with implanted devices over 5 months, not just a few days, but 5 months. During the first month, the algorithm was trained for seizure prediction. So for each patient, training was performed on that patient's seizure to try to predict the seizures in that patient. In 11 patients, an effective algorithm could be devised. So here again, in 4 patients, no algorithm could be devised to predict their seizures. In these 11 patients, the testing was performed over 4 months, providing a low, medium, and high indicator of the likelihood of seizure occurrence. And this shows how the device worked. You had subdural electrodes were implanted, electrode wires were taken to a telemetry unit, much like a VNS device, which transmitted the signal to the advisory device, which performed the computations and decided whether a seizure was likely by showing a red light or a blue light if the seizure was unlikely. These are the results for a 3-week period in one patient. This is actually a patient in whom the device worked best. You see in dark blue periods of low likelihood of seizure occurrence, in red periods of high likelihood of seizure occurrence, in white moderate likelihood, and in gray periods where data were not of acceptable quality. You can see that there are very few periods of red, and it is in fact in these periods that the 3 seizures that occurred during that time are occurring. This means that there was 100% sensitivity. The time in high advisory, the time in red was 3%, which was very low. The time in low advisory, the time in dark blue is 88%, which of course is excellent since the patient during that time is told that a seizure is unlikely, which is correct. Now, looking at the results for all the patients in that study, the results could be compiled in 10 of the 11 patients. I don't know why it was not analyzable in one of them. From these 10 patients, in one patient, the system did not perform above chance, despite the fact that an algorithm had been trained. In the testing data, it didn't behave above chance. In 9 patients, it behaved better than chance. The patients spent on average 24% of the time under high advisory, very similar to what we saw earlier. The sensitivity was 71%. That means 71% of seizures occurred during this high advisory period. The time in low advisory could not be enabled in 5 patients. In 4 of them, it ranged from 7%, which is of course quite useless because only a very small fraction of the time the patient is told is unlikely to have seizures, to the very high 88% that we saw in the previous patient. However, the clinical usefulness of seizure prediction was inconclusive. It was not clear whether the information provided to the patient was actually useful to them. In other words, whether they could make use of it to modify their behavior and their life. In conclusions, we can say that there was much enthusiasm about 20 years ago about our ability to predict seizures. Early results proved to be misguided, however, mainly because they were not correctly validated, as they did not use sufficiently long recordings primarily, and also not strong enough statistical methods. Now we understand very well the statistical validation procedure, and all the studies that have taken place since then are valid statistically. The results from many studies showed that in the fraction of patients, often about 1/3, it is possible to define a state where seizures are more likely to occur. This state may occupy 20% to 30% of the total time. Now, more recent results will be discussed by Brian Litt. There is some hope that we can do better than that at the present time. However, it's important to remember whether this type of information is of clinical utility to patients remains to demonstrate. Even if we can predict seizures better than chance, the question is, how well can we do and how useful can this be to patients? Thank you for your attention.
Thank you, Professor Gottman. So we'll move on to Brian Litt's talk. Dr. Litt is a neurologist and the director of the Penn Epilepsy Center of the University of Pennsylvania. He is the director of the Center for Neuroengineering and Therapeutics at the University of Pennsylvania. He's participated in helping bring several implantable neuro devices to the market, and he has licensed technology to NeuroPace, MC10, and NeuroVista, and has provided counsel to several companies such as Medtronic. His research lab translates neuroengineering research directly into patient care. He collaborates broadly across disciplines to invent, develop, and test new technologies and apply them to basic and clinical research. Dr. Litt's talk is entitled Seizure Prediction in 2020, Practical Challenges of Translating into Clinical Devices. Dr. Litt?
Hello, everyone. Greetings from Philadelphia. My name is Brian Litt, and it's really a pleasure to present and speak to you today at this meeting. I'm going to share my screen. And the title of my talk is Seizure Prediction in 2020, Practical Challenges to Clinical Translation. I'm a professor of neurology and neurosurgery and bioengineering at the University of Pennsylvania. These are my disclosures. Part of what I do as a professor who is half between neurology and bioengineering is that I work to bring new technologies forward into the clinical realm. And we do this with materials and implantable devices with companies like MC10. With informatics and algorithms, and there's a startup, Blackfin, that we have in Philadelphia that works in this area. And then a variety of point-of-care diagnostic and therapeutic devices, such as the hyperfine portable MRI, the NeuroPace device, which you're familiar with, and a number of other companies. None of these efforts in which we translate our work are related to this discussion and presentation today, and I received no funding for giving this lecture. The learning objectives for this lecture are to understand the components of a practical seizure prediction device, to learn the steps required for clinical translation, and to understand the biggest challenges in bringing a practical prediction device into clinical care. So the steps to getting a practical prediction device into patient use and clinical use are really, in general, these. The first is to carefully specify the indications and use cases for the device. And that's going to be key. There's a big difference between just recording and trying to predict periods of increased seizure risk and giving an alert and initiating therapy for patients for their seizures. One is much more invasive and has a harder regulatory path than the other. Engineering the device, we often focus on this, but it's just as important to think about getting it approved, your regulatory strategy, reimbursement, and adoption. So the first issue is, what is the indication? What are the use cases that you're going to use the device for? Again, seizure prediction is a really interesting intellectual exercise, but if you really want to use it to help patients, how are you going to do that in a meaningful way? So the device consists of sensors that take information, and they could be invasive or scalp or surface. They could be EEG or a number of other kinds of devices added on. There has to be data transfer to a computational device. While some computation could be done in the implantable, the work that we and others have been doing really transfer that device to a personal digital assistant or a phone, and sometimes to the cloud. Will the device just detect periods of increased risk? Will it predict probability of seizure? Will it actually inform and interact with the host or the patient? Will it advise them or actually initiate treatment like brain stimulation? These are increasingly more challenging devices to put into place. And one strategy that we've been focusing on and others is really trying to do simple things first and then adding on more difficult features as time goes on. Deciding whether or not the device will use the cloud or not is key, because there are important aspects of the practical aspects of the device related to that. So let's dig in a little bit more deeply. The first part of engineering the device, so you have your use case, say that you're going to have a device that's going to initially just inform patients of periods where they're more likely to be safe or more likely to be at increased risk. You're not going to trigger therapy, and we'll try to use that as our baseline device before we get more complicated. So the first choice is, what kind of hardware? Will you use something external like an Epitel-like device or Cerebell? Will you go sub-scalp like the UN EEG device, or like the EpiMinder device that the University of Melbourne is working on with Cochlear? Or will you go with an implantable like the Medtronic RC+S or Neurovista's device, which isn't in existence any longer, but that IP is owned by a different company? And these are important decisions. And then there are other new devices on the horizon that are coming out of academic institutions like Ricky Muller's Mini Wand device, which has many more channels and higher processing capability than an implantable. Your choice of hardware, they have to be robust, they have to be reliable. And remember, the more invasive the device is, the more the burden is for its function. For a patient to get a device implanted that's going to be invasive, it's going to have to do a lot more for them than something they wear and take on and off. You have to specify the duration. Is this only going to be worn or used during periods of increased risk? Perhaps during diagnosis or before surgery, or is it going to be worn all the time? Data quality is key. So being able to look and decide what data, what bandwidth, 1 channel, is that good enough? 8 channels? What's the quality of the data? Certainly intracranial is best, but subgaleal may be better than scalp. So important considerations. So here's an example of a data stream that comes from a subgaleal EEG device. This is 8 hours with 2 seizures. And you can see the detection algorithm here in our platform that we use in the research lab points out these 2 events. But if you're using a simple threshold detector that could operate within an implantable, you can see that you would have had a false positive here during this time, and other lower amplitude events. So simple onboard detectors, may be a little bit challenging. When you look closer at these seizure events, you can see that the actual EEG changes are here very early in the first few seconds, but then this is all scalp and movement artifact, an EMG artifact, which ends in this patient's convulsive and clonic phase of the seizure here. So looking at the data quality, what kind of data you're using for detection and prediction are going to be key, and where the electrodes are positioned makes a big difference. Here's from an intracranial device. This is a dog with natural epilepsy with the RC+S device from Medtronic used experimentally. This dog is home with an owner, and you can see these are intracranial EEG changes with seizures, which are quite robust. Our work in seizure prediction has really started with this kind of high quality intracranial data because it's a challenging computational problem. And we found that at least our approach has been try to do this on the best quality data you can get and then move to less invasive modalities as you perfect the algorithms and signal processing. What about engineering the algorithms and the interface? These are big practical challenges. There is a standard for best of kind prediction algorithms right now. An example of that is from our this article where we crowdsourced seizure prediction algorithms internationally and found ones that perform quite well, and they're freely available, the code for those. This is from 2016. But these algorithms are computationally expensive. They can't be done in an implantable, and they need to be personalized. So it turns out that if you're going to do that, they need to be periodically retrained as more data are available. The patient can and physicians can annotate their events. That you have to decide whether you're going to use these and deploy them on the cloud, intermittently update them, and then they're definitely going to need to be personalized. So this is a big challenge, the computational burden for the prediction algorithms as they're deployed, and an important decision to be made equal to the hardware. Here are just a couple of examples about algorithms that are done with local processors. So this is from the Neurovista device. This is a month's worth of data. Blue means the algorithm says likely to be seizure-free. Red predicts a seizure. White is the algorithm doesn't know, that's baseline, and gray are data dropouts. Each one of these horizontal lines is a single day. And you can see the prediction algorithm predicted each of these seizures but had a number of false positives. figuring out how to convey that information to the patient, upgrading and improving these algorithms over time is a big decision. So this device had a belt-worn computer that was recharged every night. Data had to be taken off of that from an SD card, uploaded into a large computational cluster. The algorithms were upgraded and then reprogrammed into the device on a weekly interval. So that's one strategy, but the learning is slow. Here's another example of an executable that can go onto the phone or even into the implantable. So this is a hidden Markov model, but it's done in a structure where once it's programmed in a computationally very expensive approach, it can be downloaded onto a very simple device and show baseline, pre-seizure, early onset, and ictal events. And this is work we published in the past. So key obstacles: choosing your hardware, getting your data quality, choosing your algorithms and your computational platform. What about getting approval for these devices? Well, the first is choosing the indication. So is this going to be for people who have refractory epilepsy to be worn all the time? Or is it for other people who are in the process of getting therapy titrated with initial diagnosis, for example? You have to show the device is safe, so whether it's an implant or whether it's worn externally, that it's effective, that it actually meets the indications that you've gone for, and setting the burden, the goal, like, will it predict within 30 minutes with an accuracy of 1 false positive for 24 hours? I mean, that's the example of a performance goal. It has to be reasonable and have a very low false positive rate. It's important to know that each component has to meet FDA or EU standards or wherever you're getting your device approved. So the hardware is separate from the algorithm, is separate from the patient interface and display. And so important to meet those standards. And then, is there an equivalent or a predicate device that you can compare to? So for example, if an implantable seizure device is already used for detection and stimulation, then that's an easier path to getting it approved for prediction than it is to have no existing device to go off of, and you have to do a larger clinical trial with a primary study endpoint. Finally, reimbursement, not to focus on these things, but it's important. Once you get a device approved, it doesn't mean it's going to be used. So in the United States, we have to get CMS to agree to pay for these for Medicare, and the other insurers follow. And what you need is to show that there is total value, that it improves outcome, decreases cost, and improves quality of life. It has to be equivalent or better than existing devices. Outcome, safety, cost, all issues for the insurers. But getting it approved by the insurers doesn't mean that patients are going to use it. There's going to have to be a business model, not to make a killing in the market, but to be sure that this work is self-sustainable and the device will be able to be used by medical personnel, and it has to give real value. Patients will not use it. For example, if the prediction device gives so many false positives per day that turn it off, but if it's more an advisor and just says your probability is very unlikely for seizures or very likely during this period of time, and they learn to correct it over time, improve it, and trust it, that's an example of real value. And it has to have an advantage over other technologies. For example, would you need a prediction device if an implantable brain stimulator was completely effective? Maybe not. But right now they aren't completely effective. So there's a great window for advising patients and predicting their events. So an example of a strategy could be to make a simple advisory device, one that doesn't trigger therapy and doesn't say you're absolutely going to have a seizure now, because that could give a lot of false positives, but just to do kind of a weather report for the day. That's an example of a more simple, easier bar for approval. You could use predicate hardware that already exists that you can leverage to get your data, establish algorithms that have been published and proven and vetted, and perhaps start with intracranial data where the signals are the simplest and where there's good literature. And then you can add features, you can add treatment, you can improve your precision, and maybe move to less invasive devices over time. This is just an example of a particular pathway you could use for a strategy. So that's really a whirlwind tour in 15 minutes of how you'd make a practical seizure prediction device. It really starts with, understanding what problem you're trying to solve and taking the quickest path to get something that's really useful and improves patients' quality of life. Once you've done that, you can get fancier and do more challenging and interesting and riskier things. But I think there's a direct pathway to do these things. So thank you very much for your time and attention. It's really a pleasure, and I look forward to the discussion.
Thank you, Dr. Litt, for this excellent talk. So we're right on time. So we're going to move on to the final and third talk. Dr. Elibou Wassi is an Ivado Assistant Professor in the Department of Neurosciences at the University of Montreal and a regular researcher at the University of Montreal Health Center. His research interests cover the application of machine learning techniques and the Internet of Things to improve the management of neurological and psychiatric conditions. He pursued his PhD at École Polytechnique de Montréal, during which he explored the application of artificial intelligence to intracranial EEG recordings for seizure prediction. He then joined my lab here at the CHUM in September 2018 as a postdoc researcher until a few weeks ago when he got his academic appointment. His talk is entitled Technical Considerations in Modern Seizure Prediction Approaches, Electrode Selection and Feature Engineering. Edie?
Hello, everyone. It's a great pleasure and honor to be part of the epileptic seizure prediction session, along with Professor Gottman and Professor Litt, who are two legends in the field of epilepsy research. In this short presentation, I will briefly overview some technical considerations in the field of epileptic seizure prediction, namely in terms of electrode selection and feature extraction. This program has received financial support in the form of scholarships and educational grants, and we have no conflict of interest to disclose. As discussed in the previous presentations, recent reviews on seizure prediction from our group and others have agreed on the need for continuous human intracranial EEG recordings able to capture different visual states, subject-specific assessment of the pre-ictal time, adequate identification of brain areas where pre-ictal changes can be observed or detected, new statistically significant pre-ictal biomarkers, generalized probabilistic forecasting algorithms, artificial neural networks to capture complex brain dynamics, the design of more practical forecasting algorithms which can be easily translated into clinical devices, and the rigorous statistical and pseudo-prospective testing. In my presentation today, I will be overviewing examples of techniques our groups have proposed for electrode selection or identification of brain areas with pre-ictal changes and feature extraction or design of new pre-ictal biomarkers. I will start by locating these steps within the block diagram of a traditional algorithmic seizure prediction study. It starts with data acquisition, which usually consists of intracranial EEG acquired with implanted electrodes, followed by a pre-processing step which aims to improve the quality of the signals. Electrode selection is sometimes performed to select where to implement the seizure prediction algorithm, while feature extraction aims to transform raw signals into characteristics able to display pre-ictal state dynamics. Now, feature selection is used to select the most discriminative features and use them as input to the classifier, which is a decision function. Most seizure prediction algorithms have a regularization function as a post-processing step to smooth classifier's output and improve its performance in terms of specificity. We have published a review paper in 2017 in which we explored steps along the pathway from signal acquisition to adequate performance evaluation that should be taken into account in the design of an efficient seizure prediction algorithm. We established that there is a great potential for improvement and optimization in the seizure prediction framework. Today, we'll overview examples of techniques our group has proposed for electrode selection and feature extraction. An important yet often neglected consideration for the translation of seizure prediction algorithms to implantable hardware is the practical aspects of an implantable device. More specifically, current clinical intracranial EEG consists of over 120 subdural and dot electrodes implanted to spatially sample large suspected epileptogenic brain areas. Systematic channel selection for seizure prediction, be quantitative or visual, would greatly reduce the input data size and the number of nodes in the classifier layers proportional to the number of channels, as well as allow for optimal device positioning. Channel selection remains a promising and necessary consideration for minimizing data and algorithm size, which in turn potentially optimizes the energy efficiency of miniaturized implantable seizure forecasting devices. There are several ways to make this selection. We can, for example, look at the raw data and identify those with the best signal quality. For example, those with the best signal-to-noise ratio. The electrodes can also be selected by looking at the characteristics of the signal, such as the average, the mean, the amplitude, or the prediction capabilities, such as the sensitivity or the specificity. And particularly in epilepsy, the electrodes can be selected by identifying those that are believed to be in the epileptic focus by visual inspection, or for example, by using quantitative, functional, or effective connectivity measures. Our team has mainly focused on effective or directed connectivity approaches to localize the epileptic focus. Electrodes identified within this network will be used for the implementation of the seizure prediction algorithm. One of the methods that we have used was the directed transfer function, or DTF, which is a multi-channel extension of Granger causality. In other words, a signal X is said to be causal for signal Y if previous or past values of X allows for the prediction of future values of Y. In this case, X is considered the source and Y is considered as a receptor. I won't go into the technical and mathematical details of the directed transfer function, but the results of such an analysis is a connectivity matrix whose elements reflect the causal relations between two electrodes, J and I. So if we integrate through the columns of the transfer matrix, we extract the outflow of seizure activity. And if we integrate through the rows of this transfer matrix, we quantify the inflow of seizure activity. In a previous study, we used this information of outflow and inflow as input to an unsupervised learning algorithm to adaptively identify the network of seizure activity. Here we can see an example of the results of such an analysis on seizures acquired from dogs with naturally occurring focal epilepsy. We start by isolating the frequency band of interest using a time frequency analysis. Then we compute the transfer matrix. And here we can see the outflow to inflow. And interestingly, epileptic activity generators were focal in all dogs, confirming the diagnosis of focal epilepsy in these animals, while an interhemispheric seizure activity was found in 2 out of the 3 dogs with naturally occurring focal epilepsy, what highlights the importance of electrode selection in seizure prediction. Now, in order to assess the reproducibility of the generators identified by the spectrum-weighted adaptive directed transfer function, or SWADTF, it was tested on the Melbourne Clinical Trial Seizure Database, which consists of a bit less than 3,000 seizures from 12 patients. Preliminary results are promising and shows that the spectrum-weighted adaptive directed transfer function can identify the same generator in a given patient with an average appearance of around 83%, the maximum value being around 97%. And this is a work in progress. Once electrodes have been selected, we should extract characteristics of these electrodes that can be used as inputs to a classification algorithm. In the case of cat image identification, it could be the presence of whiskers, paws, etc. But for an intracranial EEG signal, it would be characteristics such as the frequency, amplitude, or time frequency analysis. The most commonly used feature in seizure prediction remains spectral band power, which is a one-dimensional frequency analysis. While this feature characterizes the frequency decomposition of the signal, as we can see here, it cannot identify interactions between frequency components of the signal. More recently, features related to the concept of cross-frequency coupling have been shown to successfully characterize epileptic brain activity and dynamics. Cross-frequency coupling, as its name implies, can identify interactions between different frequency components of a signal. For example, here we can see the bispectrum, which is a 2D mapping of the interactions of two frequencies, F1 and F2. As shown here, much more information is retained in this higher order spectral representation of the signal than into 1D dimensional analysis on the left. Several types of cross-frequency coupling analysis exist, such as phase-amplitude coupling, in which the phase angle of a certain signal modulate the amplitude of another's. Amplitude-amplitude coupling, phase-phase coupling, which follow a similar concept. Our team began by exploring the phase-amplitude coupling in canine epilepsy. Our analysis compared pre-ictal and inter-ictal recordings, and results showed significant changes in the modulation of the amplitude of gamma waves by the phase of slow delta and theta waves. 4 combinations of frequency bands were tested, and the most significant change was between the amplitude of high gamma and the phase of delta. On the right here, we can see an example of the spatial distribution of the pre-ictal changes, and interestingly, changes were bilaterally significant in dogs with focal epilepsy, highlighting the importance of electrode selection in seizure prediction. Then we found similar results within the bispectrum, which is an advanced signal processing technique based on higher order statistics and had not been previously used for seizure prediction. Here we extracted 3 features from the bispectrum plot and found that all displayed significant changes between the pre-ictal and inter-ictal states in the same canine dataset. The figure here displays the percentage of seizures that show a significant pre-ictal change, and we can see that for this dog in particular, several combinations of electrodes and features show changes in 100% of the analyzed seizures, the dark red here. We recently extended our investigations to evaluate the possibility of characterizing different epileptic brain state, the pre-ictal state, ictal state, and post-ictal state, using 3 bispectrum extracted features on IEEG recordings from 12 patients who participated in the Melbourne University seizure prediction clinical trial. We had access to a total of 2,988 seizures, but we analyzed only those who were those longer than 30 seconds. ANOVA test shows significant changes between pre-ictal, ictal, and post-ictal recordings in at least 2 bispectrum extracted features for all 12 patients. We extracted the average magnitude and entropy of the B spectrum, and interestingly, we found that the entropy, or P1 values, decreased from the pre-ictal to the post-ictal periods in all patients. In summary, in this presentation, we overviewed examples of how electrode selection and feature extraction can be used for seizure prediction. Although the results are promising, several theoretical and technological constraints must be overcome before developing commercially available products. In the short term, the algorithms should be improved and tested in a larger number of human patients. Since these are often complex algorithms, they require robust and energy-intensive processors. So the next challenge will be to resolve the problem of hardware implementation. Then these chips should be integrated into closed-loop systems, ideally for intervention purposes. My thanks are due to our funding agencies, collaborators, as well as the whole neuroepilepsy team of the SHRM for their continuous support, and to you for your attention. Thank you.
Thank you, Elie. So what's really nice with pre-recorded presentation is that the moderator doesn't need to discipline speakers for going over there a lot of time. So we have ample time for questions. Feel free to use the Q&A function. And let's see if we have any questions. Dr. Litt, can you open your camera? Elie as well? Okay, so we have one question here from Deirdre Flort from the Canadian Epilepsy Alliance. I have epilepsy and I have a VNS device already. My question is, if an implantable device was developed, would people with a VNS device be able to use this? So I guess this is for Brian Litt.
Yeah, sure. So there are people who have VNS devices. It's not a contraindication to having other implantable devices as well. So there are people that have other brain stimulators. So I don't think that would be a contraindication at all. It's a second matter whether a prediction device or algorithm could be coupled to the VNS itself. And I think there are examples. The newer VNS device uses the EKG to trigger it. So sure, I think it's not a contraindication. You could still have it.
Here's another question I think for you. How can how can the development of hardware, algorithms, software, and clinical devices be accelerated?
That's a really great question. And I think the way it can be accelerated is by incentivizing collaboration between groups. So a number of us, John and I, and many around the world have really focused on trying to get people to share data, to share algorithms, to accelerate science, and to share hardware platforms. So, for example, the Medtronic RC plus S device is not a commercially available platform, but the US government through the NIH has really pushed making it available to investigators without cost to use it as a platform. Similarly, the Epilepsy Society and the NIH funded this crowdsourced seizure prediction competition where all the algorithms and the test data and platform all of it are available open source. So I think incentivizing groups and scientists and device makers to collaborate is the quickest way to accelerate, I think.
So here's the next question. What would you say are the top 3 currently commercially available seizure prediction devices?
I don't want to monopolize this. Sean, do you want to comment, or Ellie? Or
I mean, my impression is that there is no currently commercially available seizure prediction device. The NeuroVista that we discussed was in a clinical trial, but the company has stopped operating. I think there are lots of developments. I think there is a more fundamental issue which I wanted to bring up, which is that there are no devices at the moment because there is no good algorithm at the moment that is felt to be sufficiently general and useful to be commercialized yet. So I think that if we had a good algorithm, as Brian explained, we could probably find a way to implement it. But we haven't found, we don't really know if seizures are really predictable in sufficiently reliable way to actually make a device. So we're not there yet, is my view.
Yeah, so I'd like to comment on that too. Ellie, I don't want to, did you want to comment or?
You're on mute.
Yeah, I think Dr. Litz, your answer will be better for that.
No, fair enough. So what I would say is I agree with Jean in some ways and I disagree in others, because I think a lot has to do with how we formulate the problem. It turns out that the biggest benefit that patients in the NeuroVista trial had when they reported out was the confidence of being able to know with high likelihood of seizure-free intervals. And in some of the patients, that was quite reliable. And the one that you showed, for example, the classifiers could be trained in a way so that the probability of a seizure during the blue light was extremely low. And so I think probably the earliest devices may have some much cruder probabilistic kinds of warnings. But I think they suffer from the same challenges that a lot of our seizure localization and therapeutic devices and techniques have, which is sampling error spatially. You have to be able to find those patients where you're localized enough to get good information. So I think that it's something that, at least in the NeuroVista trial and some of the other studies we're doing now, that there are some patients where the data are quite useful and there's an opportunity. But I do agree with Jean that these algorithms are not perfected yet to the point where they could be widely applicable. I mean, another example is if you had a prediction algorithm like what was shown for the NeuroVista device, and it could trigger therapeutic stimulation. And that's the subject of a UO1 grant, a brain initiative grant that Greg Worrell and I have. You know, that may be incredibly useful and bypass the inaccuracies of the device if stimulation doesn't have any cost to it. So I think formulating the problem is going to be key to getting early devices out, but in a rigorous fashion, accurate prediction doesn't exist in the way that many people think of it when they think of prediction. Jean, did I say that okay, do you think?
Yeah, sure. I certainly have no problem with that. I completely agree that the you know, every study has found some patients in whom the system works great, and then it gives great hope that the problem seems to be theoretically solvable, because you say, well, if I can really predict seizures very well in this patient, and we assume that somehow the patients have similarity to each other, you know, they all have focal epilepsy and intractable and all these things, then why is it that it seems to fail totally in other patients, or to work very marginally in probably the majority of patients? You know, the example we all show, you showed the same example, is the one patient from Melbourne where everything worked very well, but this is one patient, and the others, nothing worked nearly as well. And so it's clear that at this point, we can't make it work in every patient. I think that it can work reasonably well, probably in a quarter to a third of the patient, maybe 40% of patients with some reasonable, you know, reasonably well. But we don't know ahead of time which patient it's going to be. And so you'd have to implant in a lot of devices, and then in many patients would be very disappointed. So it's a lot of work still.
So our next question, I think, is for Edie. Daniel Jacobs asks here, is there any consensus on what pre-ictal times could or should be?
Okay, and so that's a good question. Yeah, actually, this is still a big area of research for now. As Professor Gutman showed in the first slides of his presentation, early investigations that were looking at the few seconds or minutes previous to seizures. More recently, the American Epilepsy Society Seizure Prediction Challenge on Kaggle, they used a consensus of 1 hour preceding a seizure with a 5-minute intervention time. Although there's not like a clear standard established pre-ictal time. It's really subject specific, and it's even recent studies have shown to be seizure specific. And this is something we have recently explored also in our work with HRV features, wasn't with intracranial EEG recordings, but it could give an example. We found that the pre-ictal time was unsupervised learning was seizure specific. So each seizure, the ability to predict a seizure for this different seizures for the same patient was even different. And that's why recent studies, as presented by Professor Gutman and Professor Litt, they're talking more about seizure forecasting, a probability of high, of predicting a high seizure probability.
Okay, thanks. So here we have another question from Ian Brutton. So aside from using brainwaves for detection, what other key data elements can be used for the algorithm given external factors, medication administration, stress, sleep, and identified triggers for advising purpose to patients?
Yeah, I think it's a very good point. I mean, everybody has concentrated on brainwaves because we know that we can see seizures in brainwaves. So of course we figure that we might be able to see signs of a seizure coming in the brainwaves. And we know it comes from the brain, the seizure. So, you know, we're in the right place. But we also know that there are many other features that influence seizure occurrence, and, you know, sleep and stress, as mentioned in the question, and other variables. So I think it's hard to imagine a device that would not include the analysis of brain waves, but it's also clear that, and I think it has been demonstrated in some work from the Melbourne group, again recently, that incorporating more information about sleep stage, previous fluctuations that have occurred in you know, and things like stress, could probably enhance most likely could enhance the probability to detect seizures. It's still quite fuzzy what we need to include, but I think it's quite clear that we need to include these other variables, although I don't think that they can ever be sufficient on their own.
Just to add on, Professor Gutman, yeah, the work from Melbourne from the Melbourne group from Philippa Caroli was really interesting, in which they explored the possibility of using the circadian profile as an additional information for their classifying.
Okay. So a different type of question here from William Lowe, who maybe has watched too many spy movies, but is the risk of hacking by a third party with malicious intent into devices electronically ever a concern to the developer, especially when a cloud is utilized?
So, so if you don't mind, I'll take a crack at that. So absolutely, and I think that that's not a new concept, even just from the spy movies. You know, there was great concern when I'm blanking on his name, but one of the U.S. vice presidents had a AICD that somebody could hack it, fibrillate him. Cheney, that's right. So the device companies are very aware of this, and right now devices are not, I don't know of any patient implantables that directly connect to the internet, and they always are isolated from it and have security and very often encoding in them. And then a secondary device will offload the data and then communicate with the internet. So it's something we're dealing with right now. You know, we're working, as I mentioned, on this trial with Greg Worrell and this research implantable, where we're taking data from these dogs to a phone-like device, going up to the internet, retraining the algorithms and putting them back into the dogs. And we'll be initiating stimulation. We are doing stimulation related to prediction algorithms in them now. That grant is now moving into the next phase where we'll be doing it in people. And so the FDA and the NIH and many people share your concerns, and suffice it to say that there's a lot of security involved in this, as well as patient privacy. So I think the days of you walking into Starbucks with your implantable and having somebody hack it and, you know, do stuff to you are hopefully very far away.
I think we have time for one last question. I think this is for Dr. Gottman. So if you were 30 years younger, would you work in this field?
I guess none of the others could imagine themselves 30 years younger, but I mean, it's a, you know, if I had the knowledge, if I was 30 years younger and I had the knowledge that I had today, which of course is another story, I may or may not work in this field because I think it's an extremely difficult field, and I think that what I think I have discovered, and I wanted to bring up this question to the group, but I don't really know if there is such a thing as a pre-ictal state. You know, we always thought that there is a thing. We know there is a post-ictal state, but is there a pre-ictal state? I don't think that we know. that there is such a thing. We know that patients are sometimes more likely to have seizures because they are in some state. I mean, the patients themselves know that. They will say, when I'm stressed, I'm more likely to have a seizure. So our device says also, well, now you're more likely to have a seizure. It doesn't mean you're going to have a seizure. We're not sure. Are the changes occurring everywhere in the brain to define that pretal state? Are they localized, as Dr. Bouassi mentioned, more likely in the epileptic focus? We're not even absolutely sure of that. So it's a very difficult field, I think. What is reassuring, as I mentioned, is that we have the proper methodology to test, and we have established the proper statistics, and we know whether we can, if you can say, okay, I have an algorithm, I can test, does it work, does it not work? It works in so many patients. So we have a way of having reliable results. So this is satisfying, but it's an extremely tough field because I've always wondered, for instance, is an advisory disturbing the system such that if you tell a patient you're likely to have a seizure, it may actually not happen because you've told them. Or if you told them you're not likely to have a seizure, is the seizure more likely because you've told them? And so there's so much unknown in all this that it's really tough. Of course, if I was 30 years younger, I may still jump in this because it's so exciting, because if you could make it happen, it would be a revolution in the field of epilepsy. I didn't have time to show one of the slides that I was going to show, which shows that when patients are asked what is the most disturbing aspect of their epilepsy, there was a whole bunch of questions about medication that doesn't work, medication side effects, all kinds of things. But the top answer was unpredictability of their seizure. This is the real problem, I think, with epilepsy. So it's just a very important problem. And maybe there is some hope.
Okay, so I think we have to end our session here. We're past our time. So thank you all for attending, and thank you for our 3 speakers. Have a nice day.
Thank you.
Thank you.
Yeah.
Take care.